Yes, do you have a printed copy of the schedule? You have taken something else. I'm not sure that the whether I got the so welcome to day 2 of this meeting. And so the first session we will have 3 online talks, and it's my pleasure to invite Professor Gupta Sharma to chair this session. Okay. Hello everyone, morning and welcome to this session. So we have Anand Srivastava from ISC Bangalore online, and the title of his talk is on the slide, so I'll not just read it out. Look forward to talking to you, Anand.
Yeah. Wait, I need to figure out where my. Okay, 1 minute. Can you see my slide changes?
Yeah. Yes.
Okay. Okay. Just hold on. Now, something has happened. I don't know what. I cannot see myself and others here. I can only see my screen. Is that fine?
That's fine, I guess. Anand. Go ahead.
It has happened once with me. I gave the full talk and I later realized that I was not online. So Devayan, call me if that happens. Okay, I've kept my phone next to me.
So you can hear us, but you can't see us. Why is that?
Yes. Maybe just let me stop sharing and come back to sharing again.
So, the camera appears to be wide angle, but I don't think it covers the whole thing.
Just 1 minute. Okay, you can see my screen now. This is much better.
Yeah.
Yeah, okay, I'll get. Sorry about the confusion. So, first of all, thanks Devayan and Sandeep for inviting me. I'm really sorry I couldn't make it. Last evening, I was talking to Neha and Santosh, and it looks like it's really going great. And it would have been good to catch up with the rest of you there and kind of discuss more about all of this. But my apologies for not being able to come. Um, so what I'm going to do today is talk about more from a very higher resolution molecular interaction point of view, rather than larger uh, physical based discussion that has been taking place. And I will try to show why very fine scale molecular interactions, how they may be determining a lot of things that we see at the much larger length and time scales. Um, and the title of my talk is Low Complexity Domain Reinforces Specificity, RNA Binding Domain. Okay. Before I start, let me thank the lab. Almost all of the work that I'm going to show you has been done by Sangeeta, who is not in the picture, and also by Krishnan, who just graduated. And I have to thank all my funding agencies for providing this support. Okay, so I'll straight away jump to it. This is the only slide that is sort of introductory slide. And this I'm using to pose a few questions to all of you. Okay. And so this slide is taken from my collaborator, Sobhamai Maharana, who is currently in Biophysical Society meeting in US. And this is a very this all of you are familiar with the left hand side picture where you see all these membraneless organelles both in the nucleus and in the cytoplasm, and because the biologists have been able to identify them as separate, there has to be some distinguishing features between these droplets, and that is why they are named differently. Like you have P body, U body, stress granules, and so on and so forth. Right? Some of them have been known for ages. For example, Kahal body and others. People have been knowing about their existence for ages. For example, uh, one of the very well known marker for dementia and Parkinson disease was a Lewy body, which has been known for a while that it was connected to Parkinson, but it was only in the about 20 years, 25 years back that people connected that to alpha-synuclein, and that Lewy body was actually made up of alpha-synuclein and rich in that, and that led to uh, that was the time when it became clear that these are rich in IDPs and proteins that are of low complexity domain, which I'll talk to you more about. So that is one point. The other point is that I know the title of the meeting that Devayan and Sandeep has chosen is called liquid-liquid phase separation. But I believe the jury is still out on whether it is a purely density-density phase separation, or, and there have been recent discussions on higher order kind of organization and and theories like percolation theories, PCPC, you have theories
Can
which is,
you hear us? Can you hear me?
yes, we can.
Yes. So actually, we don't have liquid phase separation in the title. It's really biomolecular condensation. We wanted
Excellent. I
to
like that.
make that clear because I think
That's very good
so
because
that confusion is
because
not there.
you can see that there's viscosity in this, like there's a shear flow, and these are definitely, there are networks getting formed, and they eventually, based on interactions at the molecular level and other factors, they can actually also become very jellish and solid likes, and so on and so forth, and they are also separated. So it's good. I'm very happy about this. The third image that I show, a movie file, is about the organization. So these, there is, I don't remember exactly which protein. I think it's the first protein that Soba has tagged here. You can see there is a heterogeneity in the distribution of from a very high concentration to very low. So there is a overall heterogeneous organizations of proteins and RNA in these systems also. So the big questions in the field, I would say, are if you have to, like, which is not very easy and probably very formidable and impossible to answer with just theory and modeling, as we all know. But together with experiments and theory, probably there are many things that could be addressed. So the question is, if there are so many similar condensates, what makes them specific? For example, how is P body different than stress granule? Why are they localized in different regions of the cell? If it is just a physical based principles of condensate formation, then what localizes them in certain areas or certain regions of the cell? How are their compositions different? Because they have to be functionally connected with it. They are, and how is that composition determined for them? Like, are they rich in certain proteins and RNA? Do they have a certain specificity for certain RNA that they are trying to protect from degradation, or like, and also the physical properties. Like, if you are able to, let's say, probe it and look at things like viscosity or their viscoelastic properties, would they all show similar behavior, or you would say you would see differences in them? Would you see differences in them at different point of time as the cell matures or in the cell cycle? And so these are the big questions in the field, particularly from the point of view of biological functions and the intracellular organizations of them. Of course, I cannot answer all of them, but we are trying to kind of delve into some aspects of that. Okay, so just for the students in the audience also, these would be the questions that would really be valued if you are able to attempt to answering all of this. And so so I am going to discuss more about the molecular rules of phase separations for specifically for first protein, because that's the protein that we most closely work with Soba's lab. And this is also a slide that probably many of you have seen in the past. So these phase separation, these condensates that I show here, are actually composed of RNA binding. So most of the proteins that you work with in these systems are proteins that have a multi-domain characteristics with a RNA binding domain, or what is also called as RNA recognition motif, and there is a prion-like domain, or you can also call them as low complexity domain. There's subtle differences between the two, but the low complexity domains are the domains that have sequences that have frequent repeats and have and they're low complexity in the nature that the number of permutations and combinations are limited as compared to usual folded protein systems, and they are the condensation is driven by a lot of factors and also very weak interactions like cation pi become very important in all of these systems. So this is a schematics that kind of tries to show how you have on the left hand side. So this is low phase separation tendency or low condensate formation tendencies to a high condensate formation tendencies, and you can see the kind of interactions that are driving this. So you have pi interactions, which is a weak interaction but important interactions. You have a cation pi interaction from phenyl. The pi is coming, lysine is providing the cation, and then the level of cation pi's could be different. You have, for example, a tyrosine lysine will be a stronger cation pi interaction, and you could have a multiple of them combined together. So in short, what I'm trying to say is that what we see at very large scale micro or sub micro meter scales in cells are driven by these very fine interactions at the molecular level. And so I'm going to answer one particular question in this, and that is like, what is the first RNA interaction? How does and this is in particular with respect to stress granules. So this is a movie of a coarse grained molecular simulation of first molecule. And let me first explain to you, because we will be talking a lot about first molecule. You have a RRM domain, which is the RNA recognition motif domain, classically known to recognize certain RNA that the stress granule or the first would sequester it into the stress granule, for example, or in any other compartment. And on flanking on, so the pink one is the RRM domain here. Okay, and so this recently we did some work where we have actually taken the SAX profiles and any other experimental information that we have and developed this model um, to kind of show the conformational landscape that we are capturing correctly. And with Devayan, we are kind of pushing this further to bring in more subtle interactions into this with Santosh and Devayan, and also have RNA models that can interact with. But here I'm showing this movie just to give you a structural feature domain architecture of RNA of first. So the pink one is the RRM domain, and the zinc finger is the blue one that you see. And in between all of them are these different stretches of low complexity domains that are disordered proteins. So you have the SYGQ, what is classically called as the LCD domain of the first, which basically is the one that mediates phase separation. Then you have on the N-terminal side, you have the RGGs1, which we will talk about soon. And on the other flanking side of the pink module, you have the RGG2 zinc finger, RGG3, and there is towards the end there's a nuclear localization signaling sequence that actually makes sure that the first is most of the time localized in the nucleus. Okay, so briefly, this is what first structure and structure as I'm showing you here, and the modular sequence modularity looks like. Okay, and this is the data that we started working with. Okay, and this is very fascinating data to us. So if you take the first molecule and bind it with RNA, you get a binding affinity of. So this is the data from Jacob Swartz lab from Arizona University, who did a lot of work when he was with Tom Check on RNA protein interaction, and this data is particularly with first RNA interaction and a particular type of first that is used to express HNRNPA1 protein, which we are not going to discuss at all. Just know that first likes to bind to a certain RNA that will be used to express certain protein in the cytoplasm. And so, in other words, first is trying to sequester a certain RNA with itself when it is forming biomolecular condensate. And so you have wild type first, which has a binding affinity of 0.7, quite micromolar though. And the RRM part, which is thought to be the RNA recognition motif, has a poor, as compared to the full first, has a poor binding, which is 90 micromolar. And they showed that if you have RRM and on both sides are RGG1 and RGG2. So if you just take the RRM and flank it with these two RGG1 and RGG2, the binding affinity jumps from 90 micromolar to 1.9 micromolar. And to me, this is significant because it basically is telling you that the folded domain is giving you a 90 micromolar, and the disordered region, which is not thought to be very strong binders, are jumping the binding affinity by about 100 times better, or at least 50 times better here, right? And so and so these are all ITC data. So we wanted to understand what is the molecular origin of all of this. And you can think of it in terms of design principles. You can think like, suppose you wanted to make an in vitro condensate and you had a bunch of different RNA and you wanted to design a sequence that could only sequester a certain RNA with itself in the condensate. How would you go about? In if for me, this is like trying to understand the fundamental design principles behind the partitioning that may take place at synthetic level, so to speak. Okay. And to do this, what we did is we kind of did a very large scale biomolecular all atom simulations of the first protein with keep adding the with the RNA, and then you take the first RNM domain and add the flanking disordered regions into them, and then try to understand what is the interactions that are coming up and how can we understand whether we are able to explain these large increase in binding affinity. Okay. And so the first thing that we found out in this work, so this work was published last year in Journal of Biological Chemistry. The first thing we found out was that the RRM core, so you can think of this as the core of the RRM um, molecule, and they actually do not have very strong binding, as we know, 90 micromolar. So right away you can see they start dissociating, and this profile. So the way to read about this would be, this is the minimum distance between the RNA and the protein, and this shows, so the y-axis on the bottom graph, the dark orange shows the two stem of the RNA, and the middle one shows the loop of the RNA. Okay, so it's a stem loop construct, and wherever you see more brightness, those are the interacting side. The bottom is decorated with the structural motif of the first molecule, whatever we are working with. What we found out was that the moment you have a small c, the boundary residue, which is the start of the, basically you have a region around here, around 377. Those we have identified as boundary residue. The moment you have this boundary residue, you can see this as a small, transient helix here. The binding actually becomes quite stable, and you can further step. So we showed that those are the most important motifs for the protein to be bound to the RNA. And so this was very interesting because generally when you think of RRM and the LCD domains, you do not think about what is happening at the boundary. Could that be so important that if you mutate the boundary residues, the binding does not take place as strongly as it would take place with the folded part of the protein? Because generally the protein RNA interactions, you think of them getting stabilized by the folded part rather than the disordered part. So this was one finding which was quite interesting, and you can see that we took this and did further studies on this. So I'll talk about that soon. Okay.
Hear me?
Anand, can you hear us?
Yes. Yes.
Yeah, I have a question.
Yeah. Please go ahead.
So, is it possible that the RRM domain, the RNA recognition motif, is providing specificity, but additional affinity without specificity is being provided by the low complexity domains?
Both. So you are right. So actually, it's both ways. So I'll show you some data that actually. So there is a very low affinity recognition, and further stabilization is coming from the RNA. But then what we did is once it recognizes, so we have done. Let me come back to your question shortly because in interest of time, but that is exactly what we found out and answered. Yeah, yeah, that's right. So again, so let me come back to the you know. Sorry, I may not be able to get your name, so please excuse me for that. Can I know the person who was asking the question?
The extension of your name, you are Anand, I am Purnanand.
So you are complete Anand, complete bliss. I am just blissful. So sad for me. Uh, Punanand, so great question, Punanand. I'll come back to it.
Yeah,
And
Anand,
so,
one more thing. So I'm also chairing the session. So you're 19 minutes into a 30-minute talk.
okay, I will be quick. Yeah, yeah. So I'll quickly go through all of this. So then what we started looking at is what uh, you know how much does the repeat of the RGG like at, so you would see like here, right, when they had a construct when where the RGG2 part, the second, the right, the C terminal part of the protein, you can see like when they add a stretch of RGG or disordered region, you have more or less similar, experimentalists would call a similar micromolar affinity, but at some point of time there's a flipping that takes place. And so this was also quite interesting. Like this goes at the heart of like what would be the optimum length of the disordered region that you would have between, let's say, two folded regions, or like when do they start actually kicking it in? And so here we found out that when you have the larger 390, which is all the three RGGs boxes of the RGG2, then you start seeing more interactions with the RNA. And so what is happening is that there is a region that is not getting because of the structural stericity is not getting interfacing with the RNA, and that, so I can answer those questions later in the detail. So in interest of time, I'm rushing through. And then we looked at closely at the residues that are there and sort of kind of rationalize the this behavior in terms of interactions of the RGG to the further stretches of RGG are interacting better with the RNA. Okay, and because we had developed an algorithm that can cluster IDPs, which I'm not talking about today, we actually clustered the whole interaction, and we showed that there was a part of the, so you can see like there is a part that is interacting and then the part that is away from the membrane. So we could quantify all of them and rationalize how this why the longer stretches were interacting better with the RNA or not. Okay, and then this is the another interesting part we saw. So when you put both the RGG1 and RGG2 on flanking side of the RNM, you see this very interesting feature. You plot the interaction. So you subdivide the interactions of RRM with the RNA, RGG2 with the RNA, and RGG1 with the RNA. And you could immediately see that the RGG1 doesn't even bother about interacting with the, just the stem part of the RNA. It is, sorry, the loop part of the RNA. It is basically more focused on the, because it cannot, it doesn't have the space to interact with the RNA at the stem region. It actually has interaction with the RNA. So now, and we also showed that the RNA was more stable when you have the RGG1 in the system. So that essentially shows that the longer disordered region was stabilizing the RNA, and the RNA protein interactions was further stabilized by these very weak interactions but persistent interactions with the disordered region. And you can see how it forks out. Like you can see the RGG1 is on the far side of the intensity is on the far side. So they are basically clamping on the ends of the RNA, and this is on the near side of the stem loop region, the RGD2. So clearly there is a division, and that you can go back and look at the sequences and the sequence grammar of the LCD and probably get some more information out of that. Okay, so I guess I will push through, and and so then we started thinking like how do you define the, because that's the story that we are trying to say. We are trying to tell that please do take into account the regions that are considered to be the boundary residues. And then Sangeeta went through and looked at a vast set of literature. So first RNA interactions have been in play much before, much before LLPS and biomolecular condensates became, fashionable, and people started working on it. And we could see, like, so just focus on the regions, how people are defining the region between RGG and RRM. And you could clearly see that there is a huge amount of ambiguity in how would you define what makes the RRM and where does the disorder LCD part start. So you can, and so that again boils down to the point that there is something interesting happening, and you clearly don't know, like, what they could be doing. And our work in some ways tries to make the point that the boundary regions are important for binding. Okay.
I'll interrupt you for a minute here.
Yes.
Purusharth has raised his hand. He wants to ask a question.
Yes, please go ahead.
But
Raj.
since you have 5 minutes in which to wrap up and also answer questions, I'll request Purusharth to put it off till you wrap up.
Yeah. I'll wait till he finishes. No problem.
So
Yeah.
please try and finish in about.
I will. I have a couple of slides more. So then we looked at the moment you put, so if you look at the cavity of the RRM for RNA, let's say, the moment you put the C terminal region, the small stretch, you suddenly, it's like it opens up and provides like a large pocket. If you don't have that, then it doesn't have that kind of pocket. And this is the interesting part, that the transient helical motif in this domain, in the boundary region, actually provides. It provides a larger pocket for the RNA to sort of engage with the RRM. And so this is, and then we looked at all the kind of interactions that were happening, and very, very interestingly, you find that there are residues that are, for example, look at this. So the boundary region has a highly multivalent interaction. For example, the arginine 372, this is the true multivalency. You can see it is forming cation pi interaction. So it can form multiple forked cation pi or pi interactions. Sorry, not pi cation or salt bridges interactions with different residues. And so we could clearly see that this was an important hub for the RNA protein interactions. If you look at the nucleotide RNA interactions in this. Okay. And then, so for experimental purposes, we then went and mutated these TRR. So there's a small stretch called TRR there. So we mutated it to alanine. So we did, because the experiment was easy to do with triple mutant, we mutated individually and triply also. And you can immediately see the RRM. And this goes back to your question, Purnanand. The RRM RNA interaction is this is the wild type. You mutate the TRR, which is actually at the boundary residue. They are not the canonical binding site. You clearly see the pink one. I'm sorry. You see a shift in the RNA protein interaction. So the RNA protein interaction is getting weakened if you actually, so if you are able to bring the protein to the RNA, but you don't have a strong TRR interaction, it is very loosely bound to the RNA. There is a shift in the RNA protein overall distance. And similarly, we have done, looking at how that affects the RGG and RGG1 and RGG2 interactions. That doesn't seem to matter so much. So the disordered regions keep interacting the same way. The RRM actually weakens its interactions if you make the TRR as triple mutant. Okay, and that is something that Sobha Lab have been doing in their cell biological construct, where we see that the recovery time weakens when you have the first TRR interaction, which essentially tells me that the network has the recovery time actually gets better, which means that the network is kind of now more loose. And she's doing, see, they are trying to actually, because these are cell constructs and they are much difficult to interpret because there's so many other activities happening. So they are trying to do a in vitro reconstruction of first RNA with a wild type and our mutations, and trying to so I will summarize this and take up the questions later. So residue at the boundary of ordered RRM in disordered region is another feature in the RRM that we should be thinking about and thinking like how does that further improves the binding affinity. So it is like accentuating or consolidating the specificity to a stronger level. And as a multivalent interaction, I did not show the data, but we actually mutated or replaced one nucleotide of the stem region with another, and we show that the binding is lost. So there is a certain specificity to the RNA sequence and shape too. And this is what is our bigger question that we are asking with SOBA. Mutation at the boundary residue should affect the stability of RNA hairpin while RGGs continue to interact with the dsRNA. So you may not be able to sequester the RNAs that you want if you mutate these regions, and you may actually become more promiscuous in binding to different kinds of RNA. And so just I thought I should also put a future slide. So right now with Neha, who is also in the audience, and Santosh, we are trying to sort of our plan in the long term, short term, whatever you want to call it, 3 to 5 years, is to actually be able to in silico reconstitute the stress granule by doing a lot of getting data from proteomics and any kind of experimental data, so that you have some sort of in silico system where you have RNA and proteins, and then you can probe for how the properties, viscoelasticity and other properties change when you change the compositions and so on and so forth. And so for that, we are building up coarse-grained models of RNA, protein-protein interaction, protein-RNA interaction. These are some of the coarse-grained models, and currently working also with Devayan to develop a RNA model that we can couple with this and kind of study the different levels of physiological and pathological constructs in the stress granules, particularly, and P-body, because with Raj also we are been talking for a while, and that has been quite a good collaboration there too. So with this, I will end and take up questions.
Yeah, thank you, Anand. We'll just take one question from Prasad and move on to the next talk, if you don't mind.
Yeah, Poonanan, can I ask the question?
Yeah, go ahead. Very quick.
Okay. Hi, Anand. Wonderful talk.
Yes,
I was actually hoping that I'll get to meet you here because, we have not managed to meet in a while on campus. So I was looking
yes, we should
forward
be meeting soon.
Yes.
Yep.
So,
Yes.
all right. So I have a comment and a question. A quick comment that in Jacob Schwartz's data, the outlier is actually the RRM only construct, right?
Yes.
The rest of all the other constructs are kind of comparable in terms of their binding affinities, right?
So let me pull that out. So if you look at you have the RRM, and then if you add first to RGG, then you have like still a little bit similar. You can say similar, and the moment you have the RGG longer, then you start seeing the jump.
Right. So my point here was I forgot what RNA was used in this experiment.
So this RNA, so this is the stem loop RNA, and those are the RNAs, those are the mRNA for the HNRNP1, some part of the HNRP1. I have the sequence somewhere. It's in the page.
Sure, doesn't matter. My comment was that, to begin with, we don't know if this is a good substrate for RRM, right? Because despite their broad specificity to bind RNAs, there are some RNAs that RRM binds better than the others. So I'm wondering if one took a substrate which is good, which has a good binding affinity for RRM, would these numbers alter?
Yeah, you are right. So so in terms of the RNA strength of the RNA binding, this one is, I would say, this RRM would be a poorer recognition motive in terms of because it's a micromolar affinity, right?
Yeah.
And
But too high.
it's too, the values are too high, which is weak binding, right? So we found out that these are really not a very strong binder that what you would see with some other RNA, and that would be an exercise to do, like take take a RRM that has, let's say, some sort of a nanomolar or a tens of nanomolar binding, and see if at that level the RGGs and the LCDs would further accentuate it or not, right? Or they don't matter.
Yeah.
I think this is where it becomes interesting, because if they are very strong binder, then the whole thing becomes too specific anyways, right? And so the room for you probably don't need the LCDs to further consolidate the binding there.
So, thank you.
Yeah,
So-
but my
I think we'll
my-
stop there.
sorry.
We are already well out of time, so we'll.
Okay, all right. We can catch up later, Anand. Thank you.
Yeah.
Excellent. Thank you. Thank you. So we'll move on to the next talk, which is by Tanmay Paul of the University of Toronto. Phase separation of IDPs,
I'll just stop sharing.
phase separation of IDPs in analytical and computational field theoretic models. Thank you, Anand. Thanks.
Thanks, Punal.
Hi, can you see my screen?
Yes.
Yes.
Okay. Hi, everybody. I'm Tanmay. I'm at University of Toronto, and I'm going to present our group's work on phase separation of intrinsically disordered proteins using polymer field theoretic models. We are a theory and computational group at University of Toronto, and our group leader is our PI is Professor Yusan Chan, and rest of these are my collaborators in the lab. So we all know when a protein chain comes out of the ribosome, it's just a linear chain. But for some proteins, it folds into a 3D structure, and that determines its function. But for some other proteins like caprine-1, say, in the single molecule level, it doesn't fold into a 3D structure. It stays in a random coil-like structure. But in greater numbers, they can form these phase-separated droplets, which gives them functions. And this kind of phase separation is responsible for different biomolecular condensates. Some of them are nucleolase, these granules, and etc. etc. So our main aim here is to compute the phase diagrams, because the phase separation is important for those IDPs. And for this, we want to actually compute the thermodynamic equilibrium properties. And here I'm showing phase diagrams for a binary system. Here, given a temperature, the system can phase separate into two different densities. But the equilibrium condition is that for all the phases, the chemical potentials and osmotic pressure should be same. And depending on the interaction types, we can have both UCST and LCST in a single phase diagram, or a single type of UCST phase diagram. So we take a little bit different route to compute these kind of phase diagrams. We start from conventional bead spring chains as representations of proteins, where these beads represent the amino acids and springs covalent bonds. First, we write down the interaction Hamiltonian in terms of bead coordinates, but instead of solving the equations of motion for each bead, we introduce auxiliary fields, which are conjugate to fluctuating densities in the particle picture, and then construct a field picture Hamiltonian. And rest of the things how we do it with manipulating this field Hamiltonian. The advantage of doing this is that we can analytically solve some part of the theory. One of the technique is random phase approximation. And here, with this approximation, we can actually compute the free energy density analytically. And this free energy density encodes chain connectivity as well as amino acid sequence. With this analytically available free energy, we can then compute phase diagrams in minutes on a laptop. Of course, we can also solve the whole field theory, but in a lattice simulation. The corresponding technique is called field theoretic simulation, and here the fields follow this complex Lange evolution equation. And this is a typical trajectory of is a typical trajectory of this field theory simulation technique. In the panel B, the system doesn't phase separate, where these red clouds represent polymer density, but at low temperature, we see at equilibrium conditions a nice droplet. So in this technique, you can also compute average chemical potentials and osmotic pressures from which you can construct phase diagrams. But more useful things from this kind of simulation we can compute is the pair correlation functions, which are also useful for interpretation of experimental data. One of the first evidence came to show that the sequence, not only the composition of a IDP is important for phase separation, was the experiment done on DDX4 protein by the group of Julie Forman-K from our university, actually. So they showed that the wild-type variant of DDX4 phase separates in ambient conditions, but a Charles-Campbell variant of the same protein, where they just rearrange the positive and negative charges shown in red and blue, that variant doesn't phase separate. So what happens is there in the wild-type variant, the charges are blocky. These red and blue are positive and negative charges. But in the Charles-Campbell variant, they are evenly spaced. So this was one of the first evidence to show actually sequence or not the net charge that matters for phase separation. Inspired by that, people have constructed synthetic sequences. These are all 50 Mars, which have 25 positive charges and 25 negative charges. All are charge neutral, but their block sequence charge patterns are different, block sizes are different. There are two proposed measures to compute the block sizes. One of them is called sequence charge decoration parameter, SCD. And here sigma alpha is the charge of a residue at the alpha position on the sequence. So here you can see this in this parameter measure, the non-local contacts gets extra weight. So this parameter actually prioritizes non-local effects. But there is an alternate measure which is called kappa. In this measure, we scan through the sequences in blocks of 5 or 6 residues and measure the local charge asymmetry. So this measure actually prioritizes sequence local properties. We can compute phase diagrams for all these 16 sequences and determine their critical temperature, upper critical solution temperature, both in field theory simulation and random phase approximation. One of the noticeable things is that here the blocky sequences tends to phase separate more, and both analytical theory and simulation rank ordering wise matches quite well. Next, we wanted to dig deeper and wanted to see how the contact order actually affects different phase separation properties of these kind of sequences. For that, we computed the radius of gyration for these sequences in the isolated chain level at 2 different temperatures. And when we plot the RGs with SCD, they line up quite well, very well actually, for both the temperature. But for kappa, there is no visible correlation. On the other hand, this is a single chain property, of course. And we compute a multi-chain property like critical temperature, the performance of SCD and kappa both are not so good and not so bad. So from this, we can conclude that the single chain properties are mostly dominated by non-local properties of the sequences. But for multi-chain case, situation is a little bit different. It's a combination of both local and non-local effects. So what happens is there in a condensed phase, if we take a single polymer inside the condensed droplet, it mostly stays in an extended state. So here, because each bead can interact with different chains, it's okay to stay in an extended state. But at the same temperature, if you look at an isolated chain, it stays in a collapsed state because it has only itself to interact with. These kind of things, features also shows up in contact maps. In the lower half, we have isolated chain intra-chain contact maps, both in MD and FTS. Both shows highly peaked contact maps. And on the lower half, we saw the interchain contact maps in condensed phase. These are less peaked and more smeared. But another interesting thing is that the single chain contact properties also shows up in the interchain contact numbers. So this basically explains the mild correlation between single chain properties like kappa and SCD with the TCR. So next we know LLPS could be a regulator of biochemical reactions. But here we are showing that the biochemical reactions could also influence the LLPS. For example, we consider this reaction where AB neutral goes to A plus and B minus ions. These are positively and negative charge ion pairs. And in this solution, if we put a bunch of SB15, this sequence, we see an re-entrant phase separation with increasing salt concentration. But if we don't allow this as ion association, and the reaction is one way, then this re-entrance disappears. But this is not the only thing here is that the reaction rate or dissociation constant, it also gets affected by phase separation. For example, in this plot, the black curves are for polymer empty system, but the red curves are for when the system is phase separating with polymer present. Another property of LLPS or usefulness is to organize biomolecules in the condensate. One famous example is this frog molecule where F5V1 and NP1 further subcompartmentalize in a phase separated droplet or a ternary-like phase separation. So we wanted to know what could be the possible mechanisms behind this further subcompartmentalization. So we studied co-phase separation of these kind of charge sequences. We found that when two charge sequences are very similar, like SB25 and SB28, in a phase-separated droplet, they mix. But when we pair SB1 and SB28, which have like very different charge sequences, in a phase-separated droplet, they demix. So our conclusion is that this kind of amino acid sequence pattern could determine whether two proteins could demix in biocondensates. Another effect of LLPS would be regulating the dielectric environment. Once the system phase separates, we have a high concentrated region of proteins which have very low dielectric constant compared to outside where we have water. So low dielectric constant means high strength of electrostatics, but does this, we ask the question, does this affect LLPS? So it does actually. We can compute, to model this, we considered a dipole solvent where the solvent molecules can have either permanent dipole or polarizable dipole moment. And when there is no dipole moment, no nonpolar, the solvent is nonpolar, the corresponding phase diagram is this black curve. But when you turn on the polarity, the phase separation propensity actually increases. This was the effect of dielectric environment on LLPS. So we tested our theories or electrostatics-based models with an actual experiment. The experimental results came from this, again, Julie Furman's lab when lead author Taihun came. The specific system was Kaplan-1, and the main aim of this experiment was to show whether unhydrolyzed ATP affects phase separation other than providing energy through hydrolyzation. So it does actually. In the experiment, they saw that at really low ATP magnesium concentration, Kaplan-1 doesn't phase separate. At intermediate salt ATP concentration, they phase separate. At really high the droplets dissolve. But that doesn't happen for the phosphorylated variant of this caprin-1 protein, where this phosphorylated variant separates only at very low ATP magnesium concentration. So one interesting property of caprin-1 is that it has +13 charge, which almost behaves as an polyelectrolyte. But the pure caprin variant has a net charge -1, which kind of behaves as a polyampholite. So in our field theory simulation models, we model the ATP magnesium complex as a 6-bit polymer with net charge 2- and Na+ and C- as single bits. We computed the polymer protein-protein correlation functions at different ATP magnesium concentrations. So we see that at intermediate ATP concentrations, the proteins are more likely to interact with each other favorably compared to very low and very high ATP magnesium concentrations. But this non-monotonicity doesn't happen for POI caprin-1. So this basically indicates the similar reentrances as they saw in experiment. And this trend is also shown in our density snapshots from our simulations. Here, the green cloud is protein concentrations, where for caprin-1, we see this reentrance, but for POI caprin-1, no reentrance. And consistent with the experiments also, we see when there is a droplet, there is a co-localization of ATP magnesium concentration with the droplet. And this is consistent also with experimental data, which I'm not showing here, but it also shows up with our correlation function calculations also. So not only that, in experiment, they can actually compute how likely the ATP magnesium complex is to interact with a specific residue of amino acid in the caprin-1 sequence. And the green squares are the experimental data. And in our simulations, we can compute an equivalent quantity, which is an integrated correlation function. And our results are shown with red circles. So we didn't attempt to match it quantitatively, but the trends are very similar. And they can, in experiment, they can also measure the potential energy on the residue surfaces, which are shown again in the green squares. These are the experimental data. And in our simulations, we can compute potential of mean force. Actually, what we have done is that in the experiments, they introduce positive, negative, and neutral magnetic particles in the system, and they measure the potentials based on their interactions with the actual system. And in our simulations, we did a similar thing. We basically introduced very small amount of positive and negative charges in the original system, and then computed the relative difference between their positive and negative test charges, potential of mean force. And from that, we can compute a potential energy for each residue. Our results are shown again in red and orange and blue for the 3 different ATP concentrations, and the experimental data is green. Again, the trends are pretty similar, and the numbers are also not that bad either. So this exercise explains that, at least for this caprin-1 phase separation, electrostatics is enough to explain lots of things. So in summary, I just showed that only with electrostatic we can explain lots of biological situations, and in the field theory setup, we have done some work with non-electrostatics also, but I haven't shown them here yet because of the time constraint. And the take-home message here is that because the biomolecular condensate contains lots of molecules, it could be really challenging to actually simulate them in silico. But our theories can compute those phase diagrams pretty easily On the other hand, the chain representations in our theories are not very accurate compared to molecular dynamics. So in that sense, we can use these kind of theories as a complementary method in some cases. But for the cases like the Raja showed Captain 1, our theories could be almost self-sufficient. So next thing, we all know that these kind of IDPs, physiologically they operate through phase separation with this multivalent or different types of interactions, associative interactions. But when this interaction types goes awry or bad, these droplets could solidify, which then becomes pathological. And so our hope is that if we study the phase separation properties of these sequences well enough, we can come up with some effective drugs. So all our simulations, we do it with our own Python codes, and some of them are available through our lab website. You can scan this code if you want. And thank you.
Thank you, Tanmay. We've actually finished well before time, so we can take quite a few questions. There's one from Anand, I think. Yes. During the talk. Anand, are you online?
Yeah, I'm here.
Yeah, so please, yeah. Please go ahead.
You want me to ask the question, Tanmay? Good talk. Or do you want me to read it out to you?
Oh, I didn't read it. Can you ask the question, please?
So the sequences, by the way, FTS is a great model, very fast, high throughput. Of course, the specificity is not there, and that is what I'm coming to. So the moment you have hydrophobic residues, you have
Yeah,
polyelectrolyte, you have positive and negative, right? You
yeah.
don't have apolar residues into the construct.
So these theories are also, we can include those type of interactions. We have done some work on that. I just didn't present them here yet.
How would you do that? How would you incorporate the solvent accessibility or the molecular surface tension term in that?
No, so basically what I do is that we take a molecular dynamics force field,
Uh-huh.
and so we assume that the molecular dynamics force field like m pi pi or m pi pi recharged, those are
So
like good
like
enough.
course of force. Those are based like hydropathy scale kind of force field.
Not only hydropathy scale, it's a proper like 20 cos 20 matrix, and so it's not just 20 parameters, but it's like the whole matrix is given, and
Yeah, like
so
but the Tirumalai Bittencourt kind of map.
exactly, and we can incorporate those kind of interactions in our theories.
Oh, really? You have written a paper on that?
Yeah, yeah. You
Can you
can,
share? I will put my email ID. Can you email me that paper?
sure.
Okay, thanks. Very nice talk, actually. Thanks.
Thank you, Anand. Viman has a question.
Hi, Tanmay.
Hi.
Great talk. So I'm just thinking that right now the model you have presented, you have only electrostatic unit, right? That means,
Yes.
yeah, the same line of what Anand has asked. You are getting some kind of reentrant phase separation also.
Yes.
Yes. So, but it is known that that happens at high salt concentration when the electrostatic interactions are not important.
Yeah.
Exactly. That's what
Like-
happening here also actually. So because of this chemical reaction, if you increase the salt concentration to really high, most of the dissociated ion pairs would form these neutral pairs.
Okay. It's just okay. So you are supplementing something like this in the form of this. Okay, I get.
Exactly.
And can you tell me that how you get the that intermediate ATP you are getting this phase separation, but not at the high and low? What was your explanation there? I used to get it. The Caprin one.
Okay, the explanation here is that so it's Caprin 1 basically behaves as a polyelectrolyte. So for example, if you take a polyelectrolyte, highly charged polyelectrolyte, it doesn't phase separate at really small salt concentration, and at intermediate salt concentration, you can form these ion bridges.
Okay.
And those actually become mixed their enthalpic or the favorable interactions more
At
strong.
high salt concentration, there are screening.
Exactly.
I agree. Okay.
But for polyamphylite, which is POI captain 1, that doesn't happen because it's if you
I
increase the
got
salt
it.
concentration.
Thank you. Thank you.
Thank you, Vivan. So, I had a question too, Tanmay.
Yes, please.
So, is it possible to think of reentrant phase separation as being similar to uh, the effect of a macromolecular crowder that exerts an excluded volume effect like polyethylene glycol, in this sense that at very high salt concentrations, for the salt to be soluble, it engages too much of the solvent, the water, the permanent dipole solvent, and so the water is no longer available for the protein, for the macromolecule, or for the polymer, right? And so, in a sense, could this high salt reentrant phase separation be akin to the macromolecular crowding that is exerted by something that exerts an excluded volume effect?
I agree. So it's basically here, it's because of the crowding effects of this neutral ion complexes.
Thank you, Tanmay. Thank you for finishing ahead of time. It allows us to actually catch up. We're exactly at 11 o'clock and we've done 2 talks. So the third talk is by Pranay Mandal. I hope he's online. He's online.
Yeah.
Hello, Pranay.
So, can you hear me?
Yes.
we can hear.
You're from the Max Planck Institute at CBG. What is CBG?
Cell biology and genetics,
Okay, and where is that located?
It's interesting.
Dresden.
And so Pranay is going to talk to us about DNA protein co-condensation as a driving force for genome protection, function, and performance. Thank you. Yeah.
Can you share my screen? Can you see my screen?
We can share your screen, yes.
Sorry, what I meant is like, can you see my screen? Yeah. Okay. Mm,
You have to go to the full screen because we see a tiny one.
yeah. Just 1 minute. Yeah. Okay. So very good morning. It's really early morning here. So thanks to the organizer for giving me an opportunity to talk about my work. So this is my postdoctoral work that I'm currently pursuing. And today I'm going to talk about like how co-condensation of protein and DNA can lead to gene protection and shape of the nucleus. So before I move forward, I want to emphasize, like I want to spend one slide. What do you mean? What do I mean by DNA protein co-condensations? So like this is a very well-known fact that when we keep on increasing the concentration of the protein molecules, we are actually increasing the interaction parameter, and uh, that results in the liquid phase separation. That's where these red proteins actually forms a liquid droplets, and that's what we call liquid phase separations. Now on there is a different scenario where you can also tune where you can increase the interaction parameter by introducing some polymer to the system, or like polymer, by mean polymer you can you can think of DNA or RNA, where this red molecules can bind to this DNA or RNA, and as a result, what we are doing is we are tuning the interaction parameters, and as a result, it can form a similar droplet-like scenario, which in general what is called biomolecular condensates. And there are different factors behind these things, and one can give examples of like pure liquid phase separations to something of this sort of biomolecular condensates where DNA or proteins are also involved. Now this is what I call a co-condensation phenomena, because for this you need proteins which can self interact, and also you need a substrate or a DNA that can also bind to these proteins. And this is a condensation phenomena, but it needs a DNA or RNA to it. That's why we call co-condensations. Now in my talk I will stay in this regime where DNA and protein interacts to form these biomolecular condensates. Now, most of the co-condensation work that we see in literature is they are mostly concentrated like related to the inside of the nucleus, where like the different proteins can loop the DNA. For example, it can loops the enhancer region to initiate transcriptions, and also other proteins like HP1 can compact or decompact the chromatin inside that of the nucleus. Now, in today's talk, I'm not going to talk about co-condensation inside that of the nucleus, but rather I will concentrate on the nuclear periphery. Because in the nuclear periphery also, like inside of the membrane, you have you have the chromatin surface, and then you have different inner nuclear membranes which can bind to the chromatins. And so you have this DNA binding proteins, membrane proteins, and membranes in addition to the DNA. So the question that one can ask is how these different DNA and other proteins and also membranes can in self interact themselves, and how do they organize in terms of the co-condensation, or in other words, like to be specific, like can DNA protein condensation on the nuclear envelope protect and shape the nucleus? And to answer this, I am going to use a single molecular approach where we have used optical tweezer, and I will show that using optical tweezer one can show different mesoscale effect that can ultimately lead to some organelle scale level phenomena that shows that indeed DNA protein interaction can protect and shape the nucleus. So before I go forward, I want to spend this I want to explain how uh, different proteins are organized on the nuclear envelope. So if you really zoom in here, what we have is the DNA, which is shown in the yellow color. And now the chromatins are bound to a protein called BAF. Now BAF is a dimeric protein which can bind DNA to both of its sides. So as a result, it can cross-bridge DNA. And then there is this magenta protein, which is called LEM2, and this binds on the BAF. And this binds to the BAF on the one side, and on the other side, it binds to the inner nuclear envelope, membrane. So in a way, you can see these cascading structures, how chromatins are actually anchored to the inner nuclear membrane. Now, this, if you image the whole nucleus, this is also seen that these LEM proteins, which are only concentrated around the nuclear envelope, and the BAF is like they are enriched also along with the LEM2. Now, the another interesting feature of this thing is like the LEM2 can undergo liquid phase separations in vitro. So now the fact that LEM2 can binds to DNA through BAF almost provoked us to ask this question: can this combination also undergo some form of a co-condensation phenomena with DNA? So, like, is BAF DNA LEM2 co-condensation a driving force behind the chromatin organization at the nuclear periphery? Now, to answer this, I will use optical tweezer. Just a brief introduction to the optical tweezer is like you can actually, so it's a highly focused laser beam where you can trap polystyrene particles. Now, in our experiment, we can trap two polystyrene particles which are functionalized with streptavidin. And then what we can do, we can catch a single piece of DNA in between two traps. And then what one can do is to stretch or relax these DNA particles and observe the binding of the proteins, because we work in a flow chamber where we can flush different proteins in different combinations, and we can also image the system simultaneously. So really we can observe the binding kinetics, and also we can measure the force and the force that the proteins apply on this DNA. So first we wanted to really establish this binding assay protocol and wanted to check how these proteins actually bind to the DNA. So the first thing that we added was BAF. And now you can see from this, like the BAF actually binds to DNA, and you can see this cyan, the whole DNA is getting coated with these BAF molecules. And then we added LEM2 proteins, just LEM2 proteins, and you can add, as I explained, the LEM2 on itself cannot bind to DNA, and we don't see any signal on the DNA. But when we add both BAF and LEM2, and then we can see this, this is where like you can see actually the LEM2 is chemically labeled here, but BAF is also present in this case. Now here both BAF and LEM2 is binding, and that's what we are seeing here. So this actually sets the binding assay that we are going to use in throughout the experiment, and we can see that the LEM2 actually binds to DNA in a BAF dependent manner. So the second experiment that I'm going to do is, like, we catch a single piece of DNA and we will give a slack to the system. We will relax the system in presence of different proteins, and we want to observe how the system evolves. Now, before I go forward, I want to tell you how the force distance profile looks in case of naked DNA. So what you are seeing is like two beads which have a DNA in between these two beads, and what I'm doing is, like, I'm stretching and relaxing this DNA, and I'm observing how the force and the distance changes. So you can see here, like, when the distance between the two beads is very small, the force is low, which means the polymer is completely slacked and it is mainly dominated by the entropic force. And if we keep on increasing the distance, what we are doing is, like, we are stretching the naked DNA just like an elastic rubber band, and it goes into this enthalpic region. And this whole behavior can be modeled as WLC, like warm light chain model, which is shown by this black dashed line. So this is a behavior of a pure naked DNA. Now, when we relax the DNA, nothing happens. Like, I mean, we just see a slack in the DNA, and that is being reflected as a loss of signal in the DNA, because the DNA is actually going out of focus from the confocal imaging, and the DNA here is labeled with a dye through which we can see the DNA. Now, interesting thing happens when we do the same experiment with BAF. In the first case, just BAF, and the second case is BAF and LEM2. So what we are seeing is a co-condensation phenomena where you can see these droplets forming, but it happens only when DNA is relaxed. Otherwise, we don't see this droplet formation. And this is also this droplet formation is accompanied by by this plateauing of the force, where when we relax the DNA, instead of going into completely zero, the force just saturates at some force value. And this is because the extra slack that we are giving in the system actually goes into the co-condensate formation, and the system always maintain this minimum value. And depending on the protein combinations, this saturation force actually differs, and we can also find out the co-condensation energy from this. But then what we did is, like, we imaged the cell. nucleus, and what we can see is there is an enrichment of the DNA on the nuclear periphery. And so what we think is like what we are seeing this as enrichment is actually the DNA co-condensation that is happening on the nuclear periphery because of these different proteins. So as I said, like we can also measure the interaction energy, co-condensation energy from this different from this FD curve. Is like you can just integrate the area under this curve and calculate what is the energy that is stored in these co-condensates. Now, in one extreme, you have this just BAF, where the BAF actually loops this DNA and form these co-condensates, and this is happening because of the cross-bridging property of the BAF. But when you have LEM2, this is the other extreme, you have the LEM2 binding to the BAF, and this LEM2 has this IDR which can interact, and that results in an extra force, and that is also reflected in the energy calculations. Now, in between these two regimes, there exists another regime where we have mutated the IDR region, like the intrinsically disordered region of the LEM2, where LEM2 just that in that mutant, it can just bind to BAF, but it cannot self-interact. So as a result, the energy is between BAF and LEM2. So in a way, you can measure this energy and you can find what's the role of DNA binding domain, and also you can differentiate the role of protein-protein interactions from these different kinds of measurements. So the third experiment that I'm going to do is to see how stable are these condensates. In this experiment, what I'm going to do, I am going to take a preformed condensates and I'm going to stretch it. And I'm going to stretch it to unravel the DNA from the condensates. Again, I will just explain what happens when we stretch a single piece of DNA. So a single piece of DNA follows this worm-like chain model. Now, when you keep on stretching at a force of around 65 piconewton, the force plateaus out. That means like you cannot go beyond 65 piconewton, and what happens there is actually a nick is formed and a DNA starts to melt. The extra force that you are applying goes into the melting of the hydrogen bonds, and what we are seeing is a single stranded DNA as the dark region, which we are not seeing because nothing binds, like there is like we haven't put any dye that binds to single stranded DNA. That's why this region is dark, and this is getting flanked by two double stranded region. So this is the main signature that the DNA melts at the 65 piconewton if we have some nicks. Now, this is the force profile when we stretch the preformed condensates in case of BAF, which is the cyan curve, and in case of magenta, which is BAF plus LEM2. Now, what you can see, if we keep on stretching, the cyan curve actually marches the like worm-like chain model at around 40 piconewton. What it means, like, is like we can completely unravel the DNA from the BAF condensates at around 40 piconewton. But however, this magenta curve doesn't merge with this WLC line. What it means, like around this force of like 60 or 65 piconewton, we still have condensates that is not getting dissolved away. So then
There is a question. Pranay, can you hear us?
yes.
There is a question.
Yeah, so the DNA intercalating dye that you're using,
Yes.
does it have any trouble in existing within the condensate in the DNA that's wrapped up by the by BAF and LEM2?
No, so I'll come back to this question again, like, but what we have seen is like it can actually go inside the condensates.
So my question is there a difference in the quantum yield of fluorescence? Because for intercalation, you need a little bit of B-form DNA too, right? But in the condensate, it might not be in B-form.
Sorry, I can't hear you properly.
In the condensate, all of the DNA may not be in B-form, and that might affect the nature of the intercalation of the dye, and so the quantum yield of fluorescence of the dye. And so when you're seeing the fluorescence along the chain, just like you saw the dark patch for the single-stranded region where there was no dye.
Mhm.
You might see a lower intensity patch for the region where there is a condensate form. Co-condensate form.
Exactly. So the DNA may not exist in B form, but what we are using here, we are not using any dye to quantify the amount of DNA wrapped inside the co-condensate. We are just using like force distance profile, and we can fit a warm light chain model at different distance to find out how what is the amount of condensates inside the DNA, inside the co-condensates. But we are not using fluorescence profile to find out the amount of DNA, right? I mean, in this context.
But you're doing some imaging, fluorescence imaging, right?
Yes, we are, but in this experiment we are not using that.
I have a question actually.
Yeah.
At that 65 piconewton, it is also known that maybe there are some models which suggest BDNA can also go to SDN.
Yes.
Right.
So it depends on the DNA.
Yeah.
So it depends on the DNA construct. If we have a nick, it always melts at 65 piconewton. Yes, yes, yes. But it can also form sDNA, like which is like sDNA at, but it happens also at 65 piconewton.
Yeah, please go ahead.
Oh, okay, okay. So what it says is like the condensates are still are intact even if we cross 65 piconewton. And that means, like, I mean, from this force distance profile, we can actually quantify how much DNA is wrapped inside this co-condensates, and we can see that at 65 piconewton also, like almost 80% of the 75 to 80% of the DNA is still inside the co-condensate. Which means that it's very difficult to unravel the DNA from the co-condensates. And one can also calculate the energies that it takes to unravel the DNA, and it shows that to unravel one base pair of DNA from the BAF-LEM2, it takes about 4 kT of work. So this experiment shows that the DNA is actually protected inside the BAF-LEM2 condensates. Now, this experiment actually also helped us to form this hypothesis is like the DNA inside the co-condensates are prevented against melting. Like we wanted to understand how the co-condensate is actually, like what is the molecular scale that actually is helping to preserve these co-condensates and prevent it from getting melted. And what we can see here in this schematic is like when you are pulling the DNA from both the end, the force actually gets redistributed inside through this protein network, and as a result, the DNA inside is less stressed as compared to the DNA which is outside. What it means, like we wanted to check this, and this is we checked it using Cytox as an intercalating dye, and this Cytox is also spore sensor. So what we have seen is like or other people have seen is this dye is recruited more on the DNA when the DNA is more stressed. So what you can see here, like this is the yellow is the signal from the cytox, and what you can see is there is a high intensity of cytox, although cytox is present inside the co-condensate as you can see from the yellow signal, but you have high intensity of cytox outside this co-condensate, outside these magenta signals, and that shows that the DNA outside the co-condensates are stressed more. And then we wanted to check that if the melting is happening, it should happen outside the co-condensates. And this we checked by a protein called FAS. And this FAS actually is a single-stranded DNA binding protein, and that can only bind to us if there is any melting. And you can see here in this chymograph or in this snapshot that when there is a LEM2 condensate, the FAS signal is always binds away from the LEM2 condensates, and that says that when we keep on stretching, if the melting happens, it happens outside the co-condensates, and that establishes our claim that these co-condensates protect the DNA against external stress. But then the question is like, how does this translates to the organelle level? And to answer this, we did a cell biology experiment where we compress the cell to create a scenario where the chromatin is actually under tension, just like the scenario in the optical tweezer. And in this, yeah, I will wrap up now. Then what we
There's a question.
There is a question.
oh, sorry. Yeah,
Can you go to the previous slide?
yeah, this one.
Yeah, the next one. No, the next, just the
Mhm.
previous, the next one, yes. The next,
Yeah, this one.
yes. So FUS, when you are saying that it doesn't go inside the condenser, it also depends on how FUS interacts with the proteins and so on, right? Maybe
Yes.
depending on the interaction energy, maybe it's just excluding them, right? That's a possibility.
Yes, so what we have checked in, like I can show you some other slide, but what I can say is, like FUS, we have done an in vitro experiment where we have formed a LEM2 droplet and we have added FUS to it. And what we can see is LEM2 and FUS can coexist together
So that
in the absence of DNA.
Okay, thank you.
So I have a question too, Pranay.
Yes.
What exactly are you calling melting? Because classically, melting is the breaking of the hydrogen bonds to separate the strands, which is aided by the fact that you have a nick. But
Yes.
what part of it are you calling melting? Because from the diagram, it's not evident.
Yeah, so good questions. And this is also like it puzzled us for a long time. And what we are saying is melting is it can be peeling of the DNA, which is because of the nick. And that peeling is actually like we don't have any very direct proof, but what we can say is like we can see different regions, different intensities of force. Like here you can see on this edge, there is a high intensity of force, whereas in this region, there is low intensity of force. Now, this high intensity is actually because of the peeling of the DNA that happens due to a nick. And what we think is this low intensity region is actually a sDNA formation where the two strands actually separates out, where the DNA is intact, but you have only single stranded DNA which are in parallel.
You are pulling from both sides.
Yes.
You know that supercoiling of DNA can exist either as writhe or as twist. And
Yes.
so when you stretch DNA, the twist changes essentially. Instead of being 10.5 base pairs per term, it changes, right? To become less.
Yeah.
So when you have a nick, then the single stranded region allows complete rotation of the entire DNA molecule on both sides. So when
Yes.
you are exerting force pulling away, then what you will have in that single stranded region is rotation. But
Yes.
it's not very clear to me whether that rotation should also lead to breakage of hydrogen bonds.
So you are right.
Which is what melting is classically associated with. So what is causing the peeling is not evident unless the change in twist that is affected by the pulling is somehow also causing a melting.
Yes, you are right, and that also we observe. We cannot definitely say what forms of DNA, the complete, like what are the different forms of DNA at different regions in this melting region. But what I am claiming is like this FUS accumulation on the DNA can only happen because of a single-stranded DNA formation. We have control experiment where we have seen that the FUS cannot bind to B form of DNA at this condition. So whenever FUS is binding, it is some other form of DNA other than B DNA.
Say that your data is more interesting than you currently suspect.
Yes, I mean we know that.
Biochemistry websites, you'll find that this is indicating much more than people can. So the change in twist is actually also breaking hydrogen bonds. Okay. But Pranay, yeah, I'm sorry, partly I'm responsible, but
Yeah, I just have couple of slides more and I will wrap up. Yeah. So so what I was saying is like we compressed the cell and we did experiment on two types of cell. One is in control and what in the other type of cell where we have silenced LEM2. And what we can see is like we observe high DNA damage when the LEM2 is silenced. And that we think is because of this LEM2 and that happens because of the co-condensation thing. The third slide that the next slide that I'm going to show you is when what happens to the force when this LEM2 binds. Sorry, when this BAF and LEM2 protein binds. And what we have seen is like when BAF binds, the force doesn't increase. Like we hold the DNA at some 40 piconewton of force, and when BAF binds, nothing happens. But when BAF and LEM2 together binds, there is an increase in the force, and that increment in the force actually depends how stressed you are. Like more the initial tension, more is this increment in the force, and that tells me that like it is behaving some form of a springs. And now we went on to model this whole system as a springs in parallel or series, like where this DNA is a series spring which is with stiffness K1, and BAF-LEM2 come from the side and acts as a spring on parallel. And we can what it tells us is it effectively increases the stiffness of the DNA, and that is being reflected in this model. And that depends on this occupation fraction, which depends on the concentration. We went on to measure the stiffness of the DNA in presence of LEM2, and it matches very well with our model that you can see in this figure. So that means that this proteins acts as a springs. And but what does it mean in organelle scale? Now this is my last slide. So what we wanted to see is how these proteins are organized on the nuclear periphery. And you can see this it is like it forms a complete meshwork with other proteins that resides on the nuclear envelope. So we went on to model this nuclear shell, and we calculated the free energy of this. And this we did by two components. One is the bending and the stiffness component from any other proteins other than the LEM2, and this is the DNA stiffening part that is coming the contribution from the binding of these proteins. And then one can calculate the free energy. And what our model suggests is like when you silence the BAF and LEM2, the nuclear shape changes and it doesn't remain round. And this is also reflected in like we obtained cell microscopy, and you can see that the nucleus is very buckled up when BAF is silenced in this case. So I will conclude my talk here. The different single molecular approach that we showed in optical tweezer led us to formation of these co-condensates, and that can explain why we have less DNA damage inside the cells. And also we can model these interactions with the springs in parallel, and that actually controls the nuclear morphology. And at last, I want to acknowledge my PI. Like I'm a postdoc in Stephen Grill's lab, and I'm also working with Alex, who started actually this project. And for the theoretical modeling, we are working with Frank. And I also thank Ramesh and Yahoo, who are the collaborators in this work. So thanks.
Thank you, very much. Thank you to all the speakers for sticking pretty much the time. Now we just have questions.
Okay, so Pranay, I'm just regarding the model. I mean, the first model is quite nice. It's a cute model, but I mean, the second one where you actually look at when you have the Helmholtz free energy and you couple it with this kind of spring model. So here, do you know all the parameters? That's one question. How do you extract them? Of course, it seems like you're playing with the different parameters and you're seeing what kind of behavior you can see, but can you be more quantitative in this? Because currently, the way it is, I don't know if you have really quantified the shape of the cells and or the nucleus, and then how the morphology is changing. And can one do this? Like say that I change BAF or LEM concentration in the cell somehow, right? I do
Yes.
RNAi or something.
So
And
currently, that's what we are doing. And what we have quantified the amount of BAF and LEM2 inside the cell. we have also quantified the area change, like we can quantify the like surface area of this each nucleus, and we can also quantify the effective spin constant that we get from the tweezer measurement, and we also have some other AFM measurement to actually quantify these different parameters that you can see in my screens. And currently we are actually investigating that part, and really we are actually like trying to put the values to each of these parameters.
Normally, the shape of something is a sphere. When the surface tension all over the sphere is uniform, right? And what might be happening here with the knockdown, I think you said, isn't it? Knockdown
Yes.
of BAF and then two.
Yes.
Is that the whole thing is becoming non-uniform and not behaving as one unit?
Yes. So
Spherical, sphere meaning, yeah.
so the knockdown of this BAF is actually breaking this cross bridging effect, and that is actually reducing the effective surface tension on the of this nuclear shell, and that is actually you are correct. Like what we are seeing is an decrease of the effective surface tension, you can say. But we are modeling it as an like decrease in the effective tension.
Question from Ranjit and also from Biman.
Hi, Pranay, very nice talk. I have a question. So you said the 60 piconewton force is that's what's required to even partially disassemble the structure. And in cells, this would mean that the huge energy. And typically, are we aware of any mechanism to disassemble this? Because the cell has to not only assemble it, it also has to disassemble it, right? So
Yeah.
we know some ATP dependent mechanism that can disassemble that kind of energy driven process.
So what I can tell is there are measurements during mitosis where the microtubule are pulling that chromatin apart, and that pulling is actually creating some force of 100 piconewton of force. That is some cellular process, and that is beyond 65 piconewton first, and then when cells migrated through different constricted regions, there are measurements where that the force that is getting applied on the nucleus is also crossing that limit, and actually cell migration through very constricted region can actually cause DNA damage, that and there are other literatures to support this idea.
Okay, so Samantha had the same question.
Yes, I have the same question, that is it an active condensate from where the energy is coming? And my second question was, since there are multiple components in the condensate, is there any scaffold client type of interactions, like some components are very important and acting as scaffold and it can recruit clients. So do you think that fits in the scaffold client type of model?
So regarding your second questions, like it is actually you are correct, and I can see. So if you see this slide, so here these co-condensates are not actually forming due to individual protein interactions. The first protein that interacts is the BAF that cross bridges the DNA, that provides the platform for these co-condensates, because LEM2 on its own cannot bind, and then comes LEM2, and so it's a like a hierarchical order of interactions that happens, and we don't know how the internal structure of the DNA inside these co-condensates, and that will be really interesting to check. And that's your first question was if it is an active, so there is no ATP or something like that in this co-condenser, so the whole co-condenser is a passive in some sense.
Thank you.
Yeah, so Pranay, nice talk. I just wanted to know something, that the DNA you are using, is that a specifically designed one? Because BAF is specific binder or non-specific binder?
So yeah, good question, and we have checked that. So BAF is actually is present everywhere on the nuclear periphery. That actually compacts the chromatin, and that tells you inherently that there should not be any sequence dependency. I mean, in a broader sense, and that we have checked in using two or three different construct of the DNA. All the result that I showed is with lambda phase DNA, but we have also designed DNA where we have widum sequence in between that binds nucleosome, and we have seen like uniform coating all over the DNA always, and that tells me that like probably BAF is not a sequence dependent binding factor.
So one last thing is that LAM, right? The LEM. LEM2, that binding is specific.
So the LEM2 protein directly cannot bind to DNA. It only binds to DNA through BAF.
Yeah. But with BAF, it binds to a specific pocket.
So the BAF binds to the minor groove of the DNA, and what we have also quantified that like under the stretch condition, BAF binds every 6 base pair of the DNA.
Pranay, he's asking if where does LEM2 bind on BAF? Is there a particular location on BAF, like position?
Oh, okay. Yes, there is the LEM binding domain which actually binds to the dimeric BAF.
Okay. Now, what I'm asking, you are showing also experiment and also imaging data, but maybe working with a BAF which has weaker binding to LEM or something can directly tell you that this is what exactly is happening, right?
Yes.
Right? LEM and BAF interactions. I know you have other experiment to show that this is what is happening, but let's say in my system I have BAF which can interact with DNA but not with LEM2. And I do the same experiment, then the force profile should not change with the addition of LEM.
I think what he's saying is.
Yes. So in a way we have done this experiment, and like this force increase is actually due to we have other experiments to support this. Like this force increase that we see is actually due to LEM interaction through IDR. So we have a mutant which can bind to just BAF, but we don't see this force increase.
Okay. Thank you.
Yeah, very interesting, Pranay. Case of mutual crowding of BAF by DNA and DNA by BAF. Essentially, initially consolidating some kind of a condensation and then a hierarchical step involving LEM. Yeah.
That's like more like bridging. You're creating a platform and then LEM comes.
LEM comes and make your elasticity and other stuff, the interaction stronger.
Yes.
So thank you. We thank all the three speakers.
Thank you.
And wait for 3 of them. Which unfortunately you will not be able to.
Yeah.
So
You can
Send in the menu.
Okay.
We will not take the same thing. So tea coffee is in front of Ramanujan, and we will reassemble as well.